Hey, Dr. O here, and welcome to this mini lecture on blood typing. Now, we learned in A&P1 that cells are going to have a variety of things on their surfaces. They're going to have receptors so that they can receive hormones or other sort of signaling molecules. And then they're going to be punctuated with glycoproteins and uh, lipoproteins, and those can serve as signaling molecules. It's a form of communication where the cells can communicate with each other, but also with maybe other components that are going to be found in the interstitial fluid and the blood. Now, when we talk about things that are going to be on the surface of a cell when it comes to red blood cells, the thing that we're really concerned with are these chemicals that are called antigens. Now, antigens can do a number of things, but the thing that we're really concerned with are the antigens that can create an immune response. Because when we talk about taking blood from one person and giving it to another person, the last thing that we want to do is introduce a red blood cell that has an antigen that's not going to get along well with the recipient's blood. So we're concerned about these antigens that can generate an immune response. So what does this immune response look like? Well, basically it's what's called an agglutination reaction. And what that means is that the recipient's blood contains antibodies that will attach to the antigens on the surface of red blood cells and basically create these clumps of red blood cells, holding those red blood cells in place until other parts of the immune system can show up and kill these red blood cells. So that's basically what's happening. So these antigens that are capable of eliciting an immune response might also be called agglutinogens because of their ability to create this agglutination reaction, this clumping reaction. So this is the problem when we mismatch blood during a transfusion. The recipient's blood has these antibodies in it that will recognize the mismatched blood as foreign and then basically latch onto it, create these clumps, and then wait around for another aspect of the immune system to show up and do away with these red blood cells. Now, it could be a minor reaction or it could be a really bad reaction. And when it's a bad reaction, it's potentially fatal. So it's really important that we understand what's happening with this, and if we're in charge of doing blood typing, that we know what we're doing. Now, if there's one thing that you've learned probably by this point is that nothing is as straightforward as it seems. And that's certainly true when we're concerned about the antigens that are going to be on the red blood cell. Now, I know my slide says that there's at least 30 naturally occurring antigens that we can find on red blood cells. Actually, there's upwards of 250 that could be on the red blood cell. But the ones that we're concerned with are the ones that we would call antigen A and antigen B, as well as a third one called antigen D. Now, if you do a deep dive into blood typing, you're going to find that there are these minor subgroups that could also trigger an immune response in people that have some sort of a compromise in their immune system. And it would be like the Duffy antigens or the Kell or maybe even the Lewis. But we're generally not worried about those, and we're certainly not going to worry about those in this lecture. But I just wanted to tell you that, like I said, if you do the deep dive, you're going to find you're going down a rabbit hole because there are a lot of antigens that exist on a red blood cell. Some of them can create really big immune responses such as antigen A, antigen B, and antigen D. And then some of them would create only minor immune responses that in a normally otherwise healthy person we don't really need to worry about. So we're just going to concern ourselves with A, B, and D. So when it comes to the ABO blood typing, what we're looking at here are antigens A and B, and we're looking for their presence or absence on the red blood cell. And then based upon that, we're classifying the blood as either A, B, AB, or O. So what does it take to be blood type A? Well, you have the presence of antigen A and only antigen A on the red blood cell. And if you were blood type B, then that means that you have antigen B on your blood cell, only antigen B. However, if you're blood type AB, then you're going to have both antigens A and B on your red blood cells. However, if you're blood type O, then you will have neither A or B antigens on your red blood cells. Now that part's probably pretty easy to understand. The presence of the antigen is what classifies your blood. Okay? Now the complicating factor comes in when we consider what the immune system is doing. You see, during the first couple of months of life, shortly after we're born, the immune system has to up its game. And it has to start making antibodies that we have the genetic code to produce. So Mother Nature has built in a catalog of antibodies that are going to protect us from things that naturally occur in the environment. And in order to be efficient and effective at doing that and not kill ourselves in the process, the body goes around during the first couple of months and looks at our cells' surfaces and figures out what's on there so that it can learn to recognize what is self and then be able to recognize what is not self. So when the immune system shows up and is looking at your red blood cells, it's going to make a note of, do you have antigen A or antigen B, or what is the status with that? Now, whatever you don't have, the body is programmed, genetically programmed, to make an antibody for that. So what I mean is that if you have antigen A on your red blood cell but not antigen B, then your immune system is going to make an antibody against antigen B. If you have antigen B on your red blood cells but you don't have antigen A, then the immune system will make an antibody against antigen A. It's just how our genetics are. So it's not so much the presence of the antigen on the red blood cell in the recipient as much as the antibodies in the blood of the recipient, because that's where the problem arises. When we do a blood transfusion, our donor is only giving red blood cells. So when we drop those red blood cells in, we have to be worried about the antibodies in the recipient's blood reacting to those red blood cells. So here we have all of the blood types. There's 4 of them. So we have over here on the far left, we have a red blood cell with antigen A on it, which makes that type A blood. And then next to that, we have a red blood cell with antigen B on it, which makes that type B. And next to that, we have a red blood cell with both antigens A and B, which makes this blood type AB. And then the last one has neither antigen A or B, and that's type O blood. Now, if we were to have these antibodies floating around, an anti-A antibody and an anti-B antibody, if we were to expose blood type A to an anti-A antibody, that would create an agglutination reaction. So that anti-A antibody is going to latch onto that antigen A and create that agglutination reaction. The same thing would happen if we had a B-type blood cell in an area where there's the anti-B antibodies. We would have an agglutination reaction happening there. In the case of the person who has type AB blood, not only would an anti-A antibody attack it, but so would the anti-B antibody. But in the case of our friend type O blood, it doesn't have any antigen on its surface. Well, let me back up. It doesn't have antigen A or antigen B on its surface, so we don't really have to worry about any of the antibodies reacting with this. So we don't have to worry about that anti-A antibody or the anti-B antibody. So what does this mean for somebody given a particular blood type who needs to receive blood? Well, let's take a look at that. So here we have the 4 blood types, A, B, and AB and O. And the thing that we might know is that the person with blood type A will have anti-B antibodies. They won't have the anti-A because that would attack their own red blood cells, but they will make anti-B antibodies. The person with B-type blood will make anti-A antibodies, but they will not make anti-B antibodies because that'll attack the antigen B on the red blood cell. Our friend who has type AB blood will make neither anti-A or anti-B antibodies because if they made either one, it would attack their red blood cells. However, the person who is blood type O, because they don't have either one of the antigens on the red blood cell, they will make both anti-A and anti-B antibodies. So now we kind of get a clearer picture about what type of red blood cell we might be able to introduce into each one of the blood types. So let's actually do a little exercise where we do that. So here we have type A blood, has the antigen A, and we know that this person's plasma, their blood, is going to contain anti-B antibodies. And up above the blue line, we have all 4 blood types. So we're going to take a look and see which one of these types of red blood cells, or which red blood cells we could drop into a blood type A person. So we have type A blood, that's our first one there on the far left, and it has antigen A. Well, antigen A on this red blood cell right here, will that interact with our blood with the anti-B antibodies in it? No, it won't. So we can give type A blood. So the rule of thumb here is whatever your blood type is, you can take from somebody who has that similar blood type. Okay. But what about the type B blood? Well, we know type B blood has an antigen B on it, and the recipient in this case has anti-B antibodies in their plasma. So no, we cannot give this person type B blood. What about type AB blood? Could someone with type A blood take from type AB? Well, we do have this anti-B antibody right here. That could cause a reaction with that antigen right here. So yeah, the answer is, in an ideal world, we would not give this person. We would not give a type A blood person type AB blood. In an ideal world, we wouldn't do that. All right, what about type O blood? Well, type O blood doesn't have either antigen A or antigen B on it, so it can actually go here just fine with no reaction. So when we look at what we've got here, type A blood can take from type A or from type O. Let's take a look at another one. Now here we have the person with type B blood, which means that they're going to have antigen B on their red blood cells, and they're going to have anti-A antibodies in their plasma. And we know that we're going to be concerned about those anti-A antibodies. So let's take a look and see what we've got up here. So could type B take from type A? Well, no, because it has antigen A on its red blood cell, which means there would be a reaction. So no A for blood type B. But as I stated previously, type B can take from type B, so that's a go. What about from type AB? Well, in an ideal world, no, because we have the presence of antigen A on a type AB blood person. And what about type O? Well, again, there are no antigens on here, so we don't have to worry about interactions with the anti-A antibodies, so that's a good one to go as well. So type B blood can take from type B or from type O donors. Let's take a look at another one. And here we have type AB blood, and the thing that we need to be mindful of is that type AB blood is making neither anti-A antibodies or anti-B antibodies because it has antigen A and antigen B. So let's take a look at who our 4 possible donors are. Could type AB take from A? Well, yeah, it has an A antigen on it just like type A does, so like type AB does. So yeah, A would be fine. Could it take from type B? Well, type AB has a B antigen on it, so there's no antibodies to worry about. So yeah, it could take from type B. And of course, type AB can take from type AB, so that's fine. And because O would not elicit a response, even if there were antibodies floating around in the type AB blood person's blood, then yeah, we can also give type O. Now, because type AB blood can accept from all 4 blood groups, this one is known as the universal recipient. So the fact that the type AB blood person has both antigens on their red blood cell, that means that they don't have any antibodies. They have neither the anti-A or the anti-B antibodies, which means that they don't have to worry about their blood attacking any donor red blood cells. They can take from anybody. Now, our last blood type is going to be type O, and it has neither antigen A or antigen B on its cell surface, which means that this blood type will have both antibodies. It has anti-A antibodies and anti-B antibodies. So let's see who can give to O. Can A give to O? No, because type O blood has anti-A antibodies and type A blood has antigen A, so that's no good. What about type B blood? Well, blood type O has anti-B antibodies, so that's no good. What about blood type AB? Nope, there's a double no on that one because blood type AB has antigen A and antigen B, both of which would be attacked by the antibodies in blood type O's plasma. So that means that only O can give to O. Now, as we've gone through these slides, you might have noticed that O can give to everybody, which makes it a universal donor. But the ironic thing about O is that O can only take from O. It can give to everybody, but it can only take from another O. And it's because of the presence of the antibodies, the anti-A antibody and the anti-B antibody in its plasma. So now that we have an understanding of what it is we're looking at as far as the reaction, that basically we're looking at the donor's red blood cells interacting with the antibodies in the recipient's blood. That's really what we're concerned with. We now have to ask ourselves, how do we do blood typing? Well, let's say that you're in a lab and you want to go in and you want to have your blood tested. So you could take a drop of your blood and you're going to drop it into these two different serums. Now, what are in these serums? Well, one serum will have anti-A antibodies. The other one will have anti-B antibodies. Now, if you drop some of your blood into the serum with the anti-A antibodies and there's an agglutination reaction, then that tells the lab technician that this blood has antigen A. If we see the same sort of reaction with the anti-B antibodies, then that would tell us that this blood has antigen B on it. But if we don't see either reaction, then we're looking at blood that has no antigens on it, which would lead us down the path of O. So let's run through the course of this and actually look at what it might look like. So we've got our drop of blood up here, and we're going to drop it into our serum, our anti-A antibody serum. And what we would expect to see is some sort of a reaction, and we've got a reaction right there. So this is what an agglutination reaction looks like. This is a positive indicator that we have antigen A on this red blood, on these red blood cells. Okay, that's what we know so far. We still have to do the same thing with regard to the anti-B antibodies. So let's bring up a little bit more blood and let's drop it into that serum and take a look at what we got here. Now here we don't have any clumping, so there's no reaction. And that tells us that there are no antigen B, there's no antigen B on this particular blood type. So because we did have a reaction with the anti-A antibodies, but not with the anti-B antibodies, and that leads us down the pathway of saying that we have type A blood here. Let's take a look at another one. So here's our blood that we're going to test, and we're going to drop it into our anti-A antibody serum, and we get a reaction. So that tells us immediately that this blood is positive for antigen A. So we know we're at least an A. So let's take some blood and drop it into the anti-B antibody serum and see what we get here. Oh, we also got an agglutination reaction here. So that tells us that this blood is positive for the B antigen as well. So when we see this sort of a reaction, agglutination, that is the agglutination reaction with the anti-A antibodies and the anti-B antibodies when we drop our blood being tested into either one of these serums, then that tells us that what we're looking at is blood type AB. Let's look at another one. All right, so here's our blood that's being tested. We're going to drop it into the serum. We're going to see if it interacts with those anti-A antibodies, and it doesn't. So this is a no reaction. There's no clumping going on there. So we know that this one doesn't have anti-A. It might still have anti-B, or it might not have anti-B. So let's take a look. So here's our blood, drop it in the serum with the anti-B antibodies, and we end up with, again, no reaction. So because this blood did not respond to the anti-A antibodies or the anti-B antibodies, that tells us that we don't have either antigen A on this red blood cell or antigen B on this red blood cell. And that leads us to conclude that we're looking at type O blood. Now, if you were working in a lab or doing an experiment in clinic or what, some scenario where you can test blood and figure out what the blood type is, this is what it would look like. So the top one, where we have an agglutination reaction in both the serum that is anti-A antibody and anti-B antibody, that tells us that we're looking at blood type AB. If we put the blood in the two different serums and the only one that reacts is the anti-A, then that tells us that we're blood type A. If we have a reaction only in the anti-B antibody serum, then that tells us blood type B. And as we saw in our previous example, we didn't get an agglutination reaction with either anti-A or anti-B antibodies, which means that that blood type doesn't have either antigen A or antigen B, which means we're looking at blood type O. So we just talked extensively about the blood type ABO, and we learned that it is based upon either the presence or the absence of antigens A and antigen B. And when we do blood typing, we're not so much concerned with the antigens as we are with the antibodies that are going to be made in the recipient's blood, that we're going to find in the recipient's blood. And that's the reason why we have to match the correct donor red blood cells, is because of those antibodies. All right, so let's take a look at what we've got here with the Rh blood groups. Now, as is the case with this, same thing we saw with ABO, there are a lot of antigens that can qualify for this Rh reaction, but the one that we're going to be concerned with, the one that elicits the biggest reaction, the biggest immune reaction, is going to be antigen D. Now, antigen D was discovered when the scientists were doing research on rhesus monkeys. That's how that was discovered, was by doing blood typing on rhesus monkeys. And so the Rh actually comes from the name of the monkey, rhesus, because it's spelled R-H-E-S-U-S. So that's what we mean. So a positive antigen D, a positive Rh, means that you've got the presence of antigen D on the surface of your red blood cell. Most of the people in America have this. In fact, I think most of the people in the world are Rh positive. It's a pretty small minority of people that are actually Rh negative. So now that we know what is leading us down this positive or negative status, the important question is, if a person is Rh negative, does the immune system make those anti-D antibodies or those anti-Rh antibodies automatically? Does it, like with the production of anti-A and anti-B antibodies? And the answer is no, it doesn't. So it's like our genetic code only will produce anti-A antibodies or anti-B antibodies depending upon the absence of the antigen on the red blood cell. So what does that mean for antigen D? Well, antigen D will trigger an immune response in someone who doesn't have antigen D normally occurring on their red blood cells. So what that means is that someone who is Rh negative, if they have exposure to a antigen D, maybe by way of another red blood cell that has antigen D on it, then that goes through the classic pathway of triggering an immune response and the body building antibodies against it. So the same thing happens when we're exposed to a pathogen and we develop immunity to it. We get that exposure, the immune system kicks in, learns about it, develops the antibody against it, and then we harbor that antibody for pretty much the rest of our lives. So in the case of antigen D, it's a little bit different than with AB. You actually have to be exposed to antigen D in order to make an antibody. You just don't start making them right out the chute. Okay? So the most The scenario of the greatest concern when when an Rh negative person is exposed to antigen D is the scenario where you have an Rh negative mother carrying an Rh positive fetus. Because in this scenario, what can happen is a mother's immune system, if she's making those anti-D antibodies, those anti-Rh antibodies, those will attack the baby. And it can maybe be a little bit of damage, or it can be so significant that it actually leads to the death of the fetus. So this is of concern, which is why we study this. Now, we haven't talked about immunity yet and how the immune system works, but we're going to talk about it briefly right here. So the first time the body is exposed to an antigen, whether it's antigen D or whether it's some other antigen that shows up on a pathogen, the immune system says, hey, what is that? We've never seen that before. Has to take a little time to get to know it. And then it has to calculate how to put together an antibody to fight it. And then it has to build the antibody production facilities, and then it has to create a bunch of antibodies. And that takes time. So usually by the time the body is responding to the first exposure to that antigen, the immune response is a little bit on the weak side. Takes at least 10 days, and it tends to be a little bit on the weak side. But the beauty of that part of our immune system is that once we've done that, once we've recognized the foreign antigen, and once we've made the antibodies, it becomes super easy for the body to jump into gear the next time that antigen shows up, and open up the manufacturing sites and immediately start making those antibodies and basically taking care of that antigen. So whereas the first response may be a little bit weak, it could take upwards of 10 days, maybe 2 weeks before we actually get enough antibodies floating around to take care of the problem. The second one will be launched and fully in place in about 48 hours. So the problem with this isn't the first exposure to antigen D, it's the second exposure. So let's take a look at that. So before we get into looking at the Rh negative person, let's look at the Rh positive person. So what kind of blood can an Rh positive person take? We're either Rh negative or we're Rh positive, right? Well, we're not necessarily going to have any antibodies here because, we've got the antigen on the blood, so we're not going to make antibodies against our own blood. So the Rh positive person won't have any antibodies, which means that they can take either Rh negative blood or Rh positive blood, because remember, blood types can always take from their own type. But things are not this simple with the Rh negative person, so let's take a look at that. So here we have our Rh negative person, and we're going to assume that this person has not ever had exposure to antigen D, and so we don't have any antibodies floating around. All right. So when we look at this person with no anti-D antibodies, no anti-Rh antibodies, the thing that we can know right off the bat is that they'll be able to take Rh negative blood because there's no antigen on there to possibly trigger any response. But what happens when this person gets exposed to antigen D? Well, here we have our antigen D moving in, and the immune system's going to kick in, and it's going to say, look at that, there's an antigen D in here. That's foreign. Let's make some antibodies so we can kill it. So now this recipient's blood, this recipient's immune system has recognized that there was an antigen D that showed up, so now we've got the production of anti-D antibodies. First exposure might not be that problematic for the fetus if we're talking about Rh negative mother and an Rh positive fetus. But the second baby that comes along is going to have a problem, and the reason why is because of this scenario right here. So now we have an Rh negative person, an Rh negative mother who has anti-D antibodies. And if there's any exposure between the mother and the fetus with the blood, and that does happen from time to time, then the anti-D antibodies that the mother is producing is going to attack the antigen D on the fetus's red blood cells and create a problem. So let's go through a couple of scenarios looking at not only antigen A and antigen B, but now include antigen D, and see how that kind of alters our trajectory when it comes to donor and recipient. So down here in the bottom, we have blood type A positive. So we've got somebody who has antigen A and the Rh antigen on their red blood cell. So we know that this person is going to be making anti-B antibodies. So let's take a look at the first person that we have that we might receive from, and we have an A positive person. Well, we've got the rule of thumb that says like can accept from like. So certainly A positive can take from A positive, so that's a good one. What about A negative? Can an A positive person take from A negative? Well, the only antigen that we have on the blood in an A negative person is the A antigen, and that matches the A antigen that our recipient has. So yeah, this person can take from A negative. So the absence of the D antigen doesn't really adversely affect the donation process here. What about blood type AB negative? Well, we've got a little issue with that donor because we've got that B antigen. So this one is a no-go. But what about O negative? Well, an O negative person will have neither antigen A or antigen B, nor will they have the D antigen. So type O negative is going to be able to give to blood type A positive. So basically, when we look at the addition of the Rh factor, then we just have to go one more step and see what's going on there. Let's try our hand at another one. All right, so now we've got AB, and we are looking at AB negative. So we have the A antigen and the B antigen, but not the D antigen. And then we have a group of possible donors up there. So let's work our way through each one. So our first blood type is going to be A negative. So that means we're only positive for the A antigen, and we have A antigen in the recipient's blood as well. So yeah, A negative is going to be a good choice. What about B positive? Well, our recipient has B antigen on the blood, so that's not going to be a problem. But our recipient doesn't have a D antigen. And if we expose our recipient to the D antigen on the donor blood, then we're going to elicit an immune response. And that's not ideal. So we don't want to give AB negative, B positive blood. What about AB negative? Well, of course, AB negative can take from AB negative because like can always accept from like. Now, what about O positive? A little bit ago, we said that O was a universal donor. So can AB negative take from O positive? Well, normally O doesn't have any antigens on its blood, on its red blood cells, but in the case of O positive, it will bear that D antigen. And if we're trying to give that to an Rh negative person, we run the risk of creating an immune response. So there's going to be a scenario where we would not give O blood to somebody. So we could basically conclude then that O positive would not really be a universal donor, but O negative would, because O negative has neither A or B antigens, nor does it have antigen D. Now earlier we also said that AB was a universal recipient. It can take from anybody except AB negative, because AB negative has to take an Rh negative blood type. Let's take a look at one more example. So here we have AB positive. We have a red blood cell that has all the antigens that we could be possibly concerned with at this point in our careers. It has antigen A, it has your antigen B, and it has the D. Who's it going to be able to take from? Well, can it take from blood type A negative? Yeah, because this blood type AB positive has no antibodies. It will never make an anti-A antibody. It will never make an anti-B antibody, nor will it ever make an anti-D antibody. So it can take from A negative. Are we worried about that D antigen on the B positive? No, because our own recipient's blood has D antigen. So that's a good one too. What about AB negative? Oof. Well, in that scenario, we're missing the D antigen, but the absence of an antigen is not a problem. So AB positive can take from AB negative, not a problem. Now, what about this O positive? It has that D antigen on there, but so does the recipient's blood. So this one is a good donor for AB positive. So when we look at this, we can actually run through all the different scenarios of possible blood types, and the thing that we would find is that AB positive is the true universal recipient. So the true universal recipient, when we include the RH factor, is going to be AB positive. And the true universal donor, when we include RH factor, is going to be O negative. And with that, my friends, we are done with this mini lecture.